If you've ever had a cut that healed, you have seen mitosis happen in your body. Mitosis is a process of cell division in somatic cells. And soma means body, so these are pretty much all of the cells in your body, like your skin, your blood, your bones, everything except the cells that we find in our reproductive organs. The cell cycle is the life cycle of the cell, and you'll notice that it has many different phases. So after looking at the diagram in your notes packet, summarize what occurs during the cell cycle. Interphase is a preparatory phase before going through mitosis. It is the longest phase. You'll see that in this previous picture, about 75% of the time, even more than that, is going to be spent in interphase. So this is going to be a stage of preparation for division, and it's divided into 3 phases. When we look at cells under the microscope, as you can see in the picture here, when the cell is in interphase, it's going to look like a normal cell with a dark, solid nucleus. As I mentioned, interphase has 3 stages. G1 is for growth. This is when the cell is functioning normally. It's just going to continue getting bigger and carrying out its normal life processes. The S phase, S stands for synthesis. So this is when the DNA is copied, or we could say the DNA is going through replication, so that each cell will get a copy of the DNA once it divides. And G2 is getting ready for cell division. So during this time, other materials and organelles that the daughter cells will need are going to be copied. So it could be things like protein fibers in order to move the chromosomes that are usually organized by the centriole, or all of the organelles that those cells are going to need. And cells going to prepare for division. So it might start moving things around and just preparing, making sure it has everything that it needs. Mitosis is the actual division portion of the cycle. So it's a much smaller portion of the life cycle of the cell. The first stage in mitosis is prophase. So in this time, DNA is going to coil into a chromosome. A chromosome are these little X structures that you see in the nucleus of the cell, and it's just a more condensed form of DNA. So basically, it gets coiled, it gets packed together, and that's going to make it more organized so that when the DNA is being moved, it doesn't get lost or tangled or anything like that. During this time, the nuclear membrane also disappears, so a lot of times you'll see a dashed line, and the spindle fibers begin to form. And the spindle fibers are, like I said, produced by the centriole, or plants kind of have their own special structure, but those are going to help move the chromosomes once the cell starts to split into 2. During prophase, we start to see the chromosomes in the cell. So if you look at these pictures down here, you'll see little lines where the nucleus was, and those are the chromosomes. The second phase of mitosis is metaphase. So chromosomes line up at the center or equator of the cell. So you can think meta for middle. Chromosomes line up at the middle of the cell. So you'll see in these pictures here, those chromosomes have moved to the middle. During this time, spindle fibers are also going to attach to the chromosomes. And then we have anaphase. Think away, ana, away. So during this time, chromosomes are going to separate, they're going to start to move apart. And we have doubled chromosomes because of the replication during interphase. They're going to move to opposite ends or poles of the cell. So if you look in the picture, you see it kind of looks like little fingers as they are starting to separate. And then during telophase, we start to see those 2 cells. So during this time, DNA is going to uncoil. So we go from chromosomes back to chromatin, which is our relaxed form, and the nuclear membrane will reform. So during this time, we're going to have 2 nuclei. one in each almost formed cell, and the spindle fiber is broken down. So depending how late in telophase the slide is showing, if we're looking under the microscope, sometimes you'll just see kind of that finger chromosome structure, or sometimes you'll start to see what looks like a more normal solid nucleus. Finally, after telophase, we have cytokinesis. So this is when the cytoplasm is dividing, and it looks different for plant and animal cells. And for division, we're mostly going to focus on eukaryotes. In plant cells, we get what's called a cell plate that forms. So if you look, you can see this faint line between those two nuclei, and it looks different because in plants, the cell wall is starting to form. Whereas in animals, we don't have a cell wall, so the cytoplasm, we're going to make like a little protein rubber band, and it's going to squish in until those cells can pinch off and become separate. And that pinching part is called a cleavage furrow. So just a little easy way to remember the order of the phases is PMAT. So if you can remember that, then it's easy to remember that it goes prophase, metaphase, anaphase, then telophase. And I did not put interphase on here because it's important to remember that interphase is not part of mitosis. It is separate. It is how the cell is preparing for mitosis. And mitosis is the actual division of the cell. Okay, so in your notes packet, make sure you write a summary and a question, and then come get your paper stamped.
Just in case you need help, here's a rap lecture.